Welcome everyone, I'm Josemary from Molecular Cloud, and today we're meeting with University of South Wales from Australia, iGEM. If your team would please like to introduce themselves and tell us more about your project, that would be great.
So my name is Jason. I'm a third-year advanced science student majoring in genetics and microbiology. So I'm part of the wet lab team of the UNSW iGEM.
Yeah, and I'm Deborah. Oh, sorry, Sally.
You go.
Deborah, and I'm in my 4th year of biotechnology. I'm currently in the human practices team, so giving help into that team and Wiki as well.
Yeah. I'm Sayali. I'm in 3rd year doing molecular and cell bio, and yeah, I'm helping to overlook all the lab stuff.
Hi, my name is Fanez, and I'm a 3rd year medical science student majoring in human anatomy, and I'm a wet lab team member as well.
Yeah, great. So I'll just share my screen to run you guys through our project. So yeah, our project for this year is PROTECC CORAL. We're trying to save the reefs. And this is what PROTECC stands for. So prevent reactive oxygen thermal extreme caused carking. A bit of a mouthful, but carking is just Aussie slang for death. And this is our wonderful team. We've got people from a whole range of backgrounds. It's been really, really nice working with everyone from comp sci to law to science. It's been really interesting. So Deb, did you want to run us through the HP?
Oh yeah, so basically when we were thinking about what our project should be focused on, we started with thinking about bioremediation because of the really severe bushfires in the beginning of this year. And at first we were thinking about air remediation or soil remediation, something like that. But we realized that the coral reefs are going through a very severe coral bleaching event this year because of the high temperatures that are happening in summer. And it was also very relevant to us because we live in Australia and it's something that we want to protect as well. Yeah, so if you go. And yeah, so Australia's Great Barrier Reef is now experiencing its 3rd coral bleaching event. And in February 2020, it has the highest monthly temperature ever recorded on the Great Barrier Reef. So that is one of the reasons why we want to kind of combat this problem and stop coral bleaching from happening.
So just in terms of, what is coral bleaching? So what happens here is the microalgae that live in the symbiotic relationship with the coral. When you have rising temperatures, it causes stress to the coral and the algae, and then they produce these toxic reactive oxygen species, which they sort of like seep into the coral, and then the coral kick out the algae because they don't want the toxic ROS, and then that causes the corals to bleach.
I can. Oh yeah, so for human practices, this is going off our symposium slides. So during our symposium, we had Dr. Dominic Glover, which was our supervisor for the project. He went through his work on synthetic biology, and Josh McCluskey, who is a PhD student in his lab, who has helped us with wet lab development as well. But obviously they're not here today to talk about their stuff. So we'll just skip this slide. Yep, and so for human practices, we decided to kind of split our stakeholders into 6 different facets. So as you can see over here, there's traditional owners of the land. So our indigenous people, we found that since we're working with the corals on Australian land, it was very important for us to communicate whether our solution was even wanted by the indigenous people or what solutions are already in place for coral bleaching, and if they even wanted our solution. And since the traditional owners have such a strong connection with the land, we put that as our #1 priority as one of our stakeholders. And so under traditional owners, there's also biodiversity, bioprospecting, coastal protection, tourism, and commercial and recreational fishing. And those other stakeholders are also as important, but we always try to put traditional owners on top because that is our connection to Australia. Yeah.
Yep, so I'll go through the solution.
Yeah.
So this is all the wet lab stuff that we've been doing. Yeah, so these are just a few memes relevant to our project. We'll just skip that, it's okay. So basically our project has two slides to it. So we have the glutathione level, which aims to increase the level of glutathione in our algae. And the reason for this is because glutathione is a secondary antioxidant. And what that means is it's able to neutralize RAS, so the reactive oxygen species that actually caused the initial coral bleaching. And we do this by including the bifunctional glutathione synthetase gene. And this is found in Streptococcus thermophilus, which is a special bacteria. And basically, this enzyme can characterize the two-step process to make glutathione. And we're also going to increase the production of glutathione reductase, which is an enzyme that's very important as well. So if we go to the next slide, the way this works is the bifunctional glutathione synthetase is able to produce large amounts of glutathione, which the normal glutathione system is not able to do. And the reason for this is because the normal production of glutathione requires two enzymes, and one of the enzymes has strong product inhibition. And what that means is when there's a large amount of glutathione in the cell just stops producing glutathione. But the bifunctional glutathione synthetase enzyme is able to overcome that. It doesn't have this product inhibition limiting status. So basically the cell produces large amounts of glutathione, which will then which will neutralize the RAS, and also it basically turns the glutathione into oxidized glutathione. And this is what we call GSSG. So if you look at the diagram at the bottom, we have the reaction that is catalyzed by the glutathione reductase enzyme, which is able to convert glutathione disulfide back to glutathione, so the reduced version. So another aspect of our project is increasing what we call small heat shock proteins. And small heat shock proteins are what we call foldases, and they are able to hold, holdases. They're able to hold misfolded proteins, which can be due to the rising heat temperatures and prevent protein from aggregating. So protein aggregation is basically a death sentence for the cell. So if we can have large amounts of small heat shock proteins in the cell, we could prevent protein aggregation and allow the cell to survive just a bit in warmer temperatures. So the two heat shock proteins we're introducing are heat shock protein 22E and F, and these are found in a heat-loving algae called C. reinhardtii, and it's able to withstand temperatures up to 42 degrees Celsius, which is way more than our coral can withstand. Our coral can only withstand up to really 32 degrees Celsius, and coral bleaching occurs around 36 degrees Celsius. So if we can increase the heat tolerance up to 42 degrees Celsius, that means our corals will have a lower chance of bleaching. So in order to achieve this, we have a design, we propose a design for a plasmid. So we have a plasmid backbone, which is called PET19B. And within the backbone, we have what we call the lac-I promoter, which means this whole system is induced by IPTG or lactose. We also have the AMP-R gene, which gives ampicillin resistance, allows for selection through that. So basically what happens is when we induce the plasmid with IPTG, it allows the production of a transcription factor called OxyR. And what OxyR does is it floats around the cell, and when the cellular levels of RS rise above 200 nanomolars, it becomes active and it's able to bind to our bidirectional promoter, which is the yellow arrow. And what happens when that occurs, it allows the formation of the transcription machinery, and it will promote the production of both heat shock protein 22E and 22F. And this is really important because heat shock protein 2E and F form what we call dimers with each other, and these can form more complex structures which allow the whole day's activity. So in the next slide, we have some examples of our results. So basically what's shown over here is we've shown that heat shock protein 20E is able to form dimers with itself as well as form dimers with F, with heat shock 22F. So if you look over here, there's we have two gels. So in the first gel, we have two dark bands. So if you look at the orange line across, that's at 35 kilodaltons, which is the weight we expect heat shock protein 20E to run. And above that, in the blue line, we see some bands around the 60, 17, 80% imidazole, which shows around 70 kilodaltons. And we see another band over, dark band over there. And that's basically the dimer formation of heat shock protein 20E. And basically, when we saw this, we were like, oh, this is rather interesting. And this must be something new. And what we did was we tried the same gel again, but this time we added a reducing agent. And the idea of this is it will prevent dimer formation by cleaving off the cysteine residues, which are basically disulfide bonds, which can allow this dimerizing formation even after denaturing at 95 degrees Celsius. And basically in our second gel on the right, you can see how there's only a band at 35 kilodaltons and there's no band at 70 kilodaltons. Yeah, and so in our next slide, this is some preliminary modeling that's done by our dry lab team. And on the diagram on the left, there's a diagram of heat shock 22E and F forming a dimer structure. And on the right is the heat shock protein 22E. So it has this sort of donut conformation. And yeah, that's it really. So for our project, we have some acknowledgements. So of course, our supervisor, Dominic Glover and Joshua McCuskey. We also have some experts that helped us along the way. So Professor David Suggett, Dr. Sue Helen Egan, Professor Madeline Van Oppen, and Melissa Caton. Along with that, we'd like to thank the 2018 team and 2019 team acting as our mentor, and also, yeah, Revive and Restore. So here are some of our references as well. And yeah, that's basically our project in a nutshell.
That was excellent. Thank you so much for sharing that with us. So let's get started with some questions. So you mentioned the glutathione process and how you're going to use it as an antioxidant. My question is, what is the final destination for the glutathione? Are you modifying the algae to with this process, or is the goal for the coral to receive it to counteract the toxins from the microalgae?
Yeah, so we want to hit it at its source. So we're going to introduce it into the algae instead. And that makes it easier as well for us because the coral is actually an animal. So releasing GMOs as animals is like a very taboo in SynBio. So modifying the algae is a lot easier, and also it's better as a whole because it targets the source.
Okay. My next question is, you mentioned that traditional owners is a big component of your human practices. So, you mentioned you're keeping that at the forefront of consideration. So how are you doing that? Are you reaching out to those communities directly? Are you collecting feedback, or how are you gauging or measuring that feedback?
So our human practices team are currently in the process of emailing and reaching out to certain traditional owners of the land who are based in Queensland and also in New South Wales. We're trying to reach out to Indigenous academics as well, who are also scientists, and to gauge whether this might be accepted in their community. We're also trying to get in touch with a program. I don't really remember the title in my memory right now, but it basically reaches out to Indigenous communities in high schools that talk about our solution and basically educate them about synthetic biology as a whole, and also as well as our solution and what we're doing in iGEM. So those are some of the things that we're focusing on at the moment. And we're also trying to gauge how they react to, essentially a GMO being released to their land, or how they feel about this project being a hypothetical solution in the future if global warming or climate change does get to that severity where we have to use solutions like this.
I think it's great that this outreach is being done to take into consideration, how the communities would feel about it, and introducing a new GMO. It's definitely, controversial, but if, like you said, if it could have a lot of benefit to the environment and the coral, and help lower that temperature of the water, so we don't have another bleaching event, that would be definitely very helpful. Has your team thought about what success might look like? So how would you gauge the success or the improvement of lowering the temperature of the waters, or decreasing the amount of bleaching, or that sort of thing?
Sally, you want to go?
Yeah. So I think with this, if we are looking at it within the lab, we definitely want to test it within looking at coral models within a tank and seeing how they respond to this, to our solution and to our algae, and hopefully to see that even they're able to survive even with higher temperatures. And then I think if this does become something that we can implement in real life, it would similarly be replanting or sort of remediating those areas with bleached corals with new polyps, and seeing if again they're able to survive. And then alternatively, I think another thing that we're looking at is using our solution as sort of a proof of concept, in that we can engineer algae to help these corals survive these higher temperatures. And so even if it's not acceptable as a GMO, maybe we can use something like directed evolution, which could produce a similar outcome and produce the same benefits for the coral, hopefully.
Now, you've also mentioned how it might affect other species, so the overall environment. Has your team thought about ways to gauge those effects as well?
I guess,
sorry.
Sorry. For the biodiversity, we have also reached out in terms of human practices. We've also reached out to experts who are specialized in looking at the microbial communities that are present in the Great Barrier Reef, and how this can be kind of an indication on coral health. And basically, our solution, or what I've gauged from talking to these experts, is that if our solution can stop coral bleaching, or at least reduce it, then there's also a benefit to the biodiversity of the coral reef, because a lot of microbial communities live in the polyps of these corals and also the algae. So if we stop coral bleaching, then we save the coral itself and the algae that's living in the coral, and also subsequently our the microbial communities that live. So yeah, as a whole, biodiversity is conserved.
Excellent. Is there anything else your team would like our audience to know about your project, as far as initiatives or details on your methodology?
I guess what we can hope for is that we get everything done in time for the wiki freeze and all those deadlines that are coming up for iGEM. And in the future, maybe even trying to implement it in real life and actually looking at all the policies that are in place for scientific research from the Great Barrier Reef and to see how we fit in the puzzle in the future. And it's quite interesting to see the progress that we're going through. And it's also cool if we could actually bring this, or bring it to real life and talk to higher ups, I suppose, about implementing it into the Great Barrier Reef.
Yeah. And also, if you guys have any questions, feel free to contact us on unswigem2020@gmail.com.
Yeah. And also we have a
Instagram
Facebook page. Yeah, Facebook page or Instagram page as well. And it should just be UNSW iGEM. Yeah.